Welcome to Therapeutic Momentum. We take you on an inspiring journey, spanning breakthroughs in science and medicine development. Over the last century, our average life expectancy has increased by more than 20 years. Join us as we go behind the scenes with medical researchers, clinical developers, and other experts working together in the race to tackle life-altering diseases with new treatments and cures.
I'm Karen Akinsanya. I'm a translational scientist working at the intersection between biomedical research and emerging technology. Thanks for tuning in to Therapeutic Momentum. Let's get started.
Robert, it's absolutely a great pleasure to be with you today. And I'm so looking forward to this. We've known each other for a very long time, going all the way back to Merck, and we'll cover that. But let's start with you introducing yourself, telling us who you are and how you got to BMS. Maybe you can take us on that initial part of that journey, and then we'll dive into some more details.
That's great, Karen. Well, this is so fun. So I'm really looking forward to this as well. So my current role at Bristol Myers Squibb, BMS, is the Executive Vice President, Head of Research, so Chief Research Officer. And for us, that involves all the work that actually delivers investigational new drugs, INDs, from an idea generation through making the molecules and then delivering those into clinical development where they get tested for the first time. And then our group also spends a lot of time on all of the translational, biomarker, diagnostic, the laboratory work, clinical pharmacology work that supports clinical development. So that's my role today. And then working backwards from there, I've been at BMS since 2019, and I've had a couple of different roles at BMS since that time, including leading therapeutic area groups like immunology, cardiovascular, fibrosis, and global health. I came to BMS by way of the acquisition of Celgene. So I was at Celgene before BMS, and I was there for about 2 years. So I joined Celgene in 2017 until the acquisition, and at Celgene I led the immunology inflammation group. Before Celgene, I was at Merck. I led the translational medicine group, which included a computational biology, diagnostic biomarker group, but also early development as well. Before that, I was in academics for a number of years here in the Boston, Cambridge area. I had a rheumatology clinical practice. I had my own academic lab. Maybe I'll stop there at this point in time and we can go in different directions.
Well, maybe we can touch on that transition from being an academic physician scientist to industry. When you look back at that sliding door moment where I presume you would have gone on and had a continued amazing academic career, what was it that led you to actually jump into industry?
It was really an idea, and I really wanted to pursue a specific idea. And I asked myself, would it be better to pursue that idea in academics or whether it would be better to pursue that idea in industry? And I'll tell the story as if it was obvious in retrospect, and it was at the time not obvious at all. So what was the idea? So I'll go back a little bit in terms of my background as well, and this is one of the areas that we, Karen, share, I think, a passion, which is human genetics and its application to human disease. So I did my MD, PhD at Case Western, and I was in Hunt Willard's lab working on X chromosome and activation. But I was also in this amazing department focusing on understanding human genetics and human biology. And it sounds a little corny, but I kind of fell in love with human genetics. There are these questions around how much variation is there in the human genome? How much variation is there between, my chromosomes from my mom and my dad, between our chromosomes? And then you start to look across a population and how much variation is there across the entire global population? And then how does that contribute to a variety of things, including human health? So I finished my MD, PhD, went to University of California, San Francisco to do my internal medicine residency, but I was really focused on how am I going to get back to doing human genetics in the context of applying it to human health. I decided to do a postdoctoral research fellowship here in the Boston, Cambridge area with David Altshuler at what was the Whitehead Center for Genome Research. Then it became the Broad Institute of MIT and Harvard. But I wanted to continue my clinical training, so I decided to, after I got the postdoc in David's lab, to do a rheumatology clinical fellowship at Brigham and Women's Hospital. So I'm going to now fast forward about 10 years because during my time with David, practicing clinical rheumatology, and then having my own academic lab, I became more and more interested in this idea. And the idea was how human genetics could be used to make drug discovery and development a more effective, a more efficient, a more productive process. So I was doing this within the academic setting, and I worked with a number of different people from across the world to try to understand the genetic basis of diseases like rheumatoid arthritis. And one of the things that we found was that there was this overlap between the genetic discoveries in rheumatoid arthritis and approved drugs to treat rheumatoid arthritis. So that was sort of one observation. Then there was a second observation around using human genetics to predict adverse drug effects. Not you have a drug and you look for a genetic predictor of the adverse drug effect, but you have a drug against a known target, and then you take that target and say, what's the genetic variation in that target? Then you can ask, does that genetic variation predict not just efficacy, will the drug work, but also what might be potential side effects of the drug? And this was really in the early days of what's called phenome-wide association studies, or PheWAS. And we actually published the first adverse drug event, PheWAS. And it's actually a very common procedure today, but the time that we did this, it was the first time, at least to my knowledge, that this was applied to trying to predict adverse drug events for a genetically validated target. So that was sort of the second observation. And then the third was just, in general, you can use genetics, not just for rheumatoid arthritis or individual drug targets, but across the entire field to say it's more likely to predict successes and failures of drugs going through clinical development. So those three observations I was sort of making, not just within our lab, but others were making as well. And then I thought, well, where is it better to pursue this idea, human genetics and drug discovery? Is it better to stay in academics? And then I'll use a term or, but I will then transition to the term and in just a minute. Is it better to do it in academics, or is it better to do it in industry? And what I concluded was it's not an or problem, it's an and problem. So it's better to do it in academics and industry. And then I just decided, where do I want to position myself kind of along that spectrum? And it took a long time to come to this decision, probably a full year of thinking about different opportunities. But I ultimately decided to move, and that's when I joined Merck in 2013. Sorry, that was a very long answer to what was a very simple question, but hopefully that context is helpful.
Very interesting to understand how you interpreted the opportunity in genetics along the way there. So when you look back now and you think about some of the sentinel cases that have essentially validated the hypothesis that genetics can have a profound impact on how we make decisions about what to pursue when there's potential for efficacy, but also what not to pursue when there's potential for safety issues. Maybe you can walk us through that and a few examples that you've seen along the way.
Yeah. So one of the things that I'll say now, because I'll broaden it to say it's not just human genetics, it's this concept of now what I call causal human biology. And human genetics is a great example of causal human biology, but it's not the only example. So what took me from academics to industry was genetics and drug discovery. Then what got me sort of fascinated in industry and just drug discovery and development in general is the principles from human genetics can be applied in a variety of other settings as well. So that's this idea of causal human biology, knowing that a target, when perturbed, is going to have a desired outcome in human physiology that's consistent with a successful drug. And so human genetics is a great predictor of causal human biology, but there are other things as well. So what are some of the examples of human genetics and causal human biology? In my academic lab, I worked on a gene that's called TYK2, and now there is an approved drug by Bristol Myers Squibb called, the generic name is called deucravacitinib, and nib because of it inhibits the target deucra because it's a deuterated molecule. So that's how it got its name, deucravacitinib.
Oh, I didn't know that.
Yeah, it's pretty, I love the history of names and of medicines. TYK2 as a genetically validated target is one example. There are probably the poster child is PCSK9, and there were a number of early genetic discoveries. Jonathan Cohen, Helen Hobbs, a number of others as well, identified low frequency, not rare, but low frequency mutations in PCSK9. And there are multiple other examples. You can now look and see, there's so many examples now, but I think this pattern of saying causal human biology, genetics is a good predictor, and it tells you not only what the target should be, but how to modulate that target. Do you want to activate it or do you want to inhibit it? how do you want to modulate it? Is there a particular way in which you want to perturb the target? And then it also helps map out the path to clinical proof of concept and ultimately an approved drug. So PCSK9 is a good example because it's an intracellular enzyme, but it also gets secreted outside of the cell. So one of the first questions that scientists began to ask was, all right, it's a genetically validated target, but how am I going to go after it? Do I want to use a small molecule that gets inside of a cell to inhibit this particular enzyme? Or can we create antibodies that act outside of the cell to bind to and basically neutralize PCSK9? Different companies went at it in different ways. I know that I know we've talked about this in the past, but Merck went after it largely with an intracellular small molecule approach. Other companies went after it with an extracellular antibody-based approach. And it was the antibody-based approach that ultimately became approved drugs. And it was understanding not just the target, but the mechanism, and then how to recapitulate that mechanism in the form of a therapy. And people realized, that you could actually take an antibody, neutralize it, which prevented PCSK9 from binding to the LDL receptor and prevented internalization. So that is, there's more LDL receptor on the surface of cells to pull cholesterol out of the bloodstream and basically remove it for circulation. So that was the matching modality to mechanism. So I started with causal human biology, but then as I got into industry, I realized it's not just enough to nominate a target, you got to figure out how to modulate it. So that became the second principle of matching a therapeutic modality to a molecular mechanism of action, or matching modality to mechanism. And deucravacitinib was one good example of how to do that for genetically validated target TYK2. But then the antibodies against circulating PCSK9 was another good example of matching modality to mechanism. But now there's a really long list of all of this playing out, and I could certainly go through other examples, not only in our portfolio, but other approved medicines as well.
Well, here's a follow-up question. So a few of us from Merck had the pleasure of visiting Helen Hobbs, Brown and Goldstein in their lab. And it was this really exciting moment because PCSK9, as you point out, was part of a mechanism that involved LDL, control of LDL. And one of the things that I think we've come to realize as we've continued to try and exploit causal human biology and human genetics is that LDL is this amazing case because you have a circulating biomarker. Physicians in offices have access to this circulating biomarker. We have clinical trials, the 4S trial. We have all these trials that validate this endpoint as being movable by drugs in a way that leads to outcomes. I think that's been a challenge for other targets because we just don't have that wealth of data and information all the way to the physician's office. Can you speak about that and how we tackle that?
So you're actually helping me set up these kind of 5 principles that I talk about in research and development. So we've talked about the first, causal human biology. We've talked about the second, matching modality to mechanism. And you've very nicely introduced the third and the fourth, and ultimately the fifth. The third is path to clinical proof of concept. How do you know that the drug is working as predicted based upon preclinical models and genetics, et cetera, in patients with a therapeutic molecule? And in the case of circulating LDL, it was a much more straightforward therapeutic hypothesis that could be tested in early clinical development. If you could get rid of circulating PCSK9, what would happen to levels of LDL? Would they go down? If they went down, did they go down with a magnitude that would be predictive of clinical efficacy? And so the fourth principle is if you can do that early clinical experiment, path to clinical proof of concept. And the fourth principle is, how do you accelerate full development? How do you go from a proof of concept study to everything that's required for a registrational study for regulatory authorities like the FDA to say, that's a package that we can approve so that it ultimately gets commercialized and gets to people? And you're right. I mean, the reason that it was so fast for the PCSK9 inhibitors was in part because there were decades of research in epidemiology that LDL as a surrogate biomarker could predict clinical outcomes. So companies would be comfortable investing the hundreds of millions of dollars that it takes to do a registrational study and then to spend another hundreds of millions of dollars that it takes to actually look at cardiovascular outcomes because they had confidence in those principles, causal human biology around the target, matching modality to mechanism. They had early evidence, path to clinical proof of concept. But to your question, like they knew that LDL cholesterol was a very good predictor of clinical benefit in patients at a population scale because of all of the epidemiology that had done before. So that was one of the real reasons that it accelerated from the target nomination to an approved drug. And probably, I think it was at about 10 years. Yeah. But those examples, when you can tie all of those principles together, they're hard to find. We're finding more of them. Mm-hmm. So it's easy to find the targets in some ways, but it's hard to tie all the principles together. And that's what we spend a lot of our time thinking about.
Yeah. And if you go back to the original findings, even ezetimibe, right, all the way through to the most recently approved cardiovascular drugs, and our friend over at Bourbon, the team there have essentially now gone to a new modality in that space, which is very exciting. Sate Katharesan and team. This has been a 50-ish year journey.
Yeah.
So we're making, I think, really enormous strides with respect to these large chronic diseases like cardiovascular, where actually the unmet need is still high, but we understand a lot of these principles that you've just laid out. How do we go forward in other diseases where we don't have necessarily that 50-year journey of understanding the various mechanisms that are involved, getting the epidemiology, walking through these steps that it takes to fundamentally understand the link between a target and outcomes? How do we do that now?
Well, I think the roadmap is there. So I think there are examples like with PCSK9, and I know we're focusing on that as an example, but there are multiple other examples where you can lay out this roadmap. And I keep going back to the 5 principles: causal human biology, matching modality mechanism, path to clinical proof of concept. As soon as you see that, you can accelerate full development, potentially in multiple indications. And then all along the way, the 5th principle, which I haven't mentioned yet, is how do you actually ensure that patients are really benefiting in the real world? And so, we call that maximizing market access, but it's really about building the information required to say why this new medicine is going to be better than the existing medicine. So it's building out that data set. So those are the five principles. And you can see examples where all 5 principles line up really, really nicely. As I said, that's kind of rare, but if you can find it, be aggressive. But what you can also do is to say, okay, I might not have the 3rd or the 4th principle, but I see the 1st and the 2nd. And then you can begin to ask questions about, all right, what would it take to build out the necessary tools or resources or infrastructure to basically allow path to clinical proof of concept? And then you can make choices. You can say, I really believe in the target. I think we have a really good molecule to test a clinical hypothesis. And now we can take some of that risk into early development, design the right early development study. And then, it is a betting game. At the end of the day, you do have to place bets. So I think it's unrealistic to think that 100% of the ideas are going to translate into new medicines. But if you go from currently what is about 10% of ideas that start in early development become new medicines, if you can double or triple that from 10% to 20 to 30%, you're moving in the right direction. So I think we take the principles, we use history to say, here's the lesson about how we can apply them. And then you do your best to sort of build out the data set to support the ideas to increase probability of success through clinical development. And it doesn't have to be 100%, but if you can, again, double or triple it, that's success. And then we iterate. Every step along the way, we're going to have better tools to build out, I think, the necessary infrastructure to sort of support those principles. And we're just going to get better and better and better over time. And I think we're starting to see that play out.
Yeah. So the metrics of how long it takes and how much it costs are things that we have to live with every single day. How do we now balance the pressure for speed with the need for this rigorous validation? And when is it worth slowing down? Any examples, any thoughts on that? Because I think, we say patients are waiting. We all feel a sense of urgency every single morning when we wake up to go figure out, how to speed things up, how to interrogate these difficult questions. But I guess the question is, how do we balance that pressure and speed?
So I think it's worth just pausing for a moment on this concept of target crowding and why speed is so important relative to target crowding. So it used to be that the first approved medicine against a validated target, the 2nd or the 3rd medicine that came after it would be several years behind. So there was a several year head start. Now that time has shrunk to several months, in some cases even almost simultaneous submissions. So more and more companies are going after the exact same targets, and they're doing it faster and faster. I saw a statistic recently that said that of the number of targets that are being prosecuted across the entire industry, about 2% of the targets are being pursued in 25% of the clinical trials.
Right.
So it's the target crowding around a small number of targets is massive, which means the competition continues to increase. So that's why speed is so important. The other component of this is if you're first or second to market, there's a big drop off in terms of commercial success if you're third, fourth, or fifth. And so at some point, if you're fourth or fifth and you're not really differentiated, there's not much value that you're delivering to patients, and therefore there's not much value that you're getting as a return in terms of market value.
Not a great use of resources.
So it's not great use of resources.
Yeah.
Exactly.
And
exactly.
focus, yeah.
So that target crowding and speed is really important. So then there's this tension, which you're setting up nicely, which is how much do you emphasize speed versus quality? Because if you're best in class, that tends to be the most successful profile overall. But if you're best in class and third, fourth, or fifth, or too far behind, you're not going to help patients. So we talk about this as quality is the first and most important kind of anchoring point, but urgency and efficiency, so QUE, is really important. And so, yes, speed is important, but if you don't anchor on quality, I think you're going to make tough decisions along the way. At the same time, if you only focus on quality and don't focus on urgency and efficiency, you're going to be, you may be best, but you're going to be too far behind and patients aren't going to benefit. So if you anchor on quality, but you emphasize the importance of urgency, then you have to do it efficiently because we need to continuously drive down costs. So there's not a great formula for this. It does depend upon any individual program. And then what I like to say is it also goes back to those 5 principles. If you really believe in the target and you have a great molecule, it may be worth spending more time in early development to get past the clinical proof of concept if you really believe and you're working through, I think, some fundamental issues. So it's worth taking that time to anchor on quality, potentially at the expense of urgency. But then if you get it right, and by getting it right, it's dose, it's schedule, it's getting the right indication, maybe there's a patient subset. If you get it right, now you're ready to accelerate full development, to really position the medicine to benefit the most number of patients. So when you apply that speed factor, that urgency factor, it's really important. I've seen mistakes where people apply the speed kind of factor, the urgency factor too early, and you don't work through fundamental issues in early development of dose and schedule. I've also seen it apply too late, where you spend too much time trying to get it like precisely right, and then you're just too far behind. So it's a tough problem of getting quality, urgency, and efficiency all right, but that's what makes the job so fun.
It surely does. So let's now talk about urgency, acceleration, and technology.
Yeah.
All of what we've spoken about so far is really built on data and what I'm now calling data and decision science, right? How do we make better decisions? You've just gone through some wonderful algorithms for how you do that. What role does technology have? There's a lot of excitement right now about technology. I'll say the word AI. There's tons of enthusiasm here, but where are we really? And what are you seeing today? And what are you looking forward to in the future with respect to how technology helps us with the quality, the urgency piece?
Yeah. So one, I think that often people will take a new technology and then go looking for a problem to solve. And often they think they're solving a problem that's really important, and it turns out not to be a very important problem. So they solve it, but you're like, turns out that wasn't the most important problem to solve. So I keep going back to those 5 principles: causal human biology, matching modality mechanism, path to clinical proof of concept, accelerating full development, maximizing market access. And then I say, okay, how do we do that, preserving quality, urgency, and efficiency? And where can this new technology help us kind of in that broader kind of spectrum of concepts, principles, and ideas? And then now you have a problem that you're trying to solve, and you can ask, is the technology suited to solve this particular problem, which is really fundamental to advancing a new medicine. And increasingly, especially with some of the tools that we're talking about, including data, artificial intelligence, machine learning, et cetera, like those tools are solving the fundamental problems. But occasionally you say, all right, those tools are solving a different problem that isn't on critical path. So defining the problem statement is really important, asking what tools are available to help us solve those problems, and then being very specific with the applications that you apply to those technologies in the context of those problems. So for example, data, there is a lot of translational data that helps inform us on some of the decisions. So in some instances, it could be a shared passion of ours, human genetics, and the ability to use human genetics from biobanks, probably less relevant in clinical trials, but more population scale biobanks. So now you're trying to solve a problem to pick better targets, that first principle, that makes a ton of sense. You could also say in the context of clinical studies, using kind of translational data sets to help us understand causal human biology, or to make sure the medicine is working the way that you think it is, path to clinical proof of concept. And then you could ask, okay, how will the data sets inform us on that particular problem statement? And I think there are examples where we're doing that well, but there are also examples where we try to generate too much translational data in our clinical trials, and suddenly you're then compromising urgency and efficiency without really solving the problem. So I think that's a good example of saying, yes, generating data is really important, but if it occurs at the expense of quality, urgency, and efficiency, we probably shouldn't do it. And that's always kind of a source of discussion within our own clinical teams, because we all want more data, but it can't compromise the fundamental problem of getting medicines to patients faster. So we could probably go in a lot of different directions on this topic, including different analytical tools like AI to help us with interpreting the data.
Yeah. I mean, AI certainly, I think, is having quite an impact on the efficiency with which we deal with documents and text, and a lot of fundamental pieces that become a huge assist to the endeavor of filing and all of these pieces. I think that's low-hanging fruit.
Yep.
There's higher hanging fruit though, right? Which is predictive power. I think one of the hopes is that AI and machine learning allow us to predict outcome. Well, we've just talked about the journey to understanding outcome for LDL, PCSK9, and what an incredibly long, complicated, multi-pronged journey that was. What is your view now of where we are with respect to predictive power from computation, machine learning, and the sort of myriad of pieces that are required to allow us to get to that point where we have predictive power?
All right. So my kids often will ask me a question, and then I'll give them a very long answer. They're like, Dad, it was kind of a yes-no question, and you're still going on and on. So they'll call that an LBL, a long, boring lecture. So I try to make it a bit more focused. This is going to be, Karen, a little bit of a long, boring lecture, because I want to kind of build out a few things within here. So I will apologize not only to you, the listeners, but also my kids for potentially making a simple question a long, boring.
Not sure how simple it is, but I think we'll get there.
All right. So number one is what I might be sort of thinking about is the Gartner hype cycle. You've got this peak of sort of hype. You've got this trough of disillusionment. Then you've got this slope of enlightenment before you get to a plateau. So I think we often talk about AI in sort of a binary way. Where is it to helping, something, and it's like, it's a yes or no. It's helping us or it's not. But I actually think of all these along kind of that hype cycle. And I think there are things like, how to use large language models to interpret, text, and to help us with writing, document writing, engaging in regulatory interactions, answering questions, et cetera. Like for that, we're very clearly on this path to enlightenment. But there are other parts of the drug discovery process where it kind of in earlier stages, and in some cases, the hype cycle, in some cases, the trough of disillusionment. And if you think just how many steps there are in making a medicine, some people say there are like 1,000 different steps. You can apply that hype cycle to each of those 1,000 different steps. In some places, we're farther along. In other cases, we're really early. So I think areas that we're on kind of the early part of that slope of enlightenment. And by the way, it's a long, steep slope for some of these things. So I think people also anticipate if we're on that slope, that the slope is going to hit the plateau within a year or two. But I think it's a long slope for many things. But I think one area in particular that I'm very excited about is the ability to match modality to mechanism and make molecules faster. So we call that predictive molecule invention. I think we're doing a lot in that particular area. I think we're kind of earlier, and I wouldn't say we're kind of in the trough of disillusionment, but we're very early on a very sort of slow slope of enlightenment in causal human biology. And, why is that? I think, there are probably a couple of things. One, just biology is hard. Human biology is just hard. We've got a lot of good information, but it's still just very complex and very, very difficult. And we often train models that then help us with these correlative insights into human biology, but they don't help us with the causal insights into human biology. And why is that? Because I still think we're trying to figure out truth in causal human biology. And so these models work really well when you can train them on a truth that you have confidence in. They don't work particularly well, or they'll give you, I think, misleading information if you train them on these correlative data sets. And so that's kind of where we are right now. We're training a lot of these big models on very interesting correlative data sets, but we're not getting at the real root of the problem, which is causal human biology. We're on the right path, and I think they're going to continue to help. But I think we're kind of really early on that slope of enlightenment. And I also think that slope is a very gradual slope. So we're farther along the hype cycle for things like predictive molecule invention. We're on the slope, but it's really early and it's shallow for things like causal human biology. At the same time, where we are today, it's going to be the worst that it ever is in human history. Or say it a different way, it's only going to get better. So we do need to invest in this. We just need to be kind of realistic about how long it's going to take in order to get to that sort of plateau where we all want to be.
Well, I enjoyed your LBL. I thought it was great. So, yeah, I would say I agree with everything you've just said. I want to go back to, I think it was the mid-90s when we talked about sequencing the human genome and what an incredible opportunity that was, and it was accomplished. And I think at that time we felt that, and I'd say lay people read headlines that said, now we've got the sequence of the entire genome, we're going to solve disease. I feel like those moments keep happening. I think we've heard headlines that now we have AI, we're going to solve 100 diseases in 10 years, we're going to, revolutionize this very difficult thing we do, which is drug discovery and development. I think your point is well taken, that there are areas where revolution is happening. But some of the hardest questions we have to answer, we're barely scratching the surface. And if we think about precision medicine, that's another one, right? There was a moment, and I think we have precision medicines that are super successful and have been very impactful for patients. But context is everything. And your point about truth is so well stated, because we have certain truths, especially when we get to the end of this journey, and then we have, real world outcomes. There's a lot of truth because we've built it. But when we come to a patient who has a disease where we have no drugs, we have very little insight into why that patient is sick, or even to your point about gene dose response, maybe someone on a certain point in that gene dose response is sick, but a person slightly further down isn't. Now, why is that? What's happening in the cell? What's happening at the level of the protein? We have so little truth about all of that that I think it's very hard to imagine, and in a very short-term way anyway, that AI is going to impact that. Now, maybe it helps us to interrogate those situations by running more experiments and doing them more efficiently. But I agree with you. I think that those very hard questions that we haven't got answers to yet, it will take time for these technologies to have an impact.
Yeah. And what I love about where we are in terms of science, and I know we're kind of in this interesting time in the world right now, but what I love about where we are is there are some really big ideas that are out there.
Yes.
And we have more tools at our disposal to answer those big ideas than ever before. And the unmet medical need, in this case, the need for new medicines, it's as large as it's ever been. So I think all of the sort of the fundamentals are there. And so what I love is to be able to say, all right, what is that big idea? And then, what are the individual modules or the catalytic steps that are required to get towards that big idea? In some cases, we'll get to the big idea faster than we think. That doesn't happen very often. I think more likely it's going to be we get to the big idea, over time. So there are kind of two quick examples of this and you mentioned one, kind of genetic dose response curve. So when I was kind of in the, 2008, 10, 12 kind of range, we began to see genetic variants that were associated with traits, both common and rare. And then the idea is, could you use that to predict validated targets for drug discovery? And kind of there was this idea that we had at the time that if you had just not one variant, but multiple variants, and multiple variants that perturb the target in different ways, in some cases gain or loss of functions, you could have this allelic series, that allelic series could be used to predict the kind of efficacy and toxicity profiles, and therefore, really do a much more effective job at nominating targets very early in a drug discovery journey. And so while those concepts have been true, I think it's been slower than we expected to get that allelic series, because as we've sequenced more and more genes, it's just the effect size of many of these are very small, which means the power of a genetic study has to be very, very large. So I think the idea is playing out in terms of allelic series. It's taken a lot longer than I expected. We're kind of 10, 15 years in, and I was sort of hoping we'd have an allelic series for every single gene in the human genome. We're not there, but I think we're making kind of progress. I think another example, I had the privilege of working with Zach Kahani and others in this thing called I2B2, Informatics for Integrating Biology in the Bedside. So here in the Boston Harvard system. And, we wanted to try to use AI, machine learning, to try to describe phenotypes from electronic medical records, embed human genetics for some of the predictors. And, at the time, we just didn't have truth to try to get at the human phenotypes. And so we spent a lot of time trying to do just basically manual chart reviews to say, all right, here's truth, now train the model, but the data sets were just too small. So the big idea was, can we use electronic medical records to help us with all the things that we talked about earlier in terms of FWAS, et cetera. And I think we're making progress towards that big idea. It's been slower than expected. But one area where it's been faster than expected has been over the last couple of years with the large language models to basically extract text to say, all right, here's something that is actually a prediction. So in some cases, that slope can be very, very slow. And then at some point you hit this catalytic moment where it really takes off. And I think at least in that specific example, the large language models to extract really relevant information from text, that's been a catalytic moment. But we're still working towards the big problem, which is how do we incorporate everything for drug discovery and development?
Yeah. And I want to make sure that people understand that while we are unpacking some of the challenge, I think we're both very excited about another tool in our toolbox, another tool in the armamentarium that helps us. I think from this perspective of image analysis, language models, these are very powerful. I think there was a great example of work that Google had done early on with respect to retinopathy, where it literally took the blindfolds off to some degree because the machine learning model could bin people who had certain disease states just based on a retinal scan. An individual couldn't do that. And that is incredibly powerful, right? The question of why it was sort of binning people became a sort of dilemma, which I'm not sure if they've solved yet, but maybe they have. So taking the blindfolds off and extending the reach of human capability, I think, is exciting.
Yeah.
It's just knowing when to apply it.
Yeah,
right?
and all right, that's a great example, because I think there are examples in imaging, digital pathology, and other places where we say, all right, we know truth. We also know that our ability to get at truth through manual interventions and interpretation is probably too slow and efficient, but we can at least define models based upon truth, and now those models are incredibly powerful. And it's a great example of, I think, AI having a very real near-term impact. In other cases, like root cause of disease, like what is that truth? Like we just don't know. And I think it's asking way too much of these models to be able to say it's going to uncover the secrets of human history, to say, all right, we're going to help use these models to understand the root causes of human disease. I just don't believe that that's possible yet, because we don't know what truth is. Now, as we make progress towards defining the nodes of that sort of overall model, I think then we can begin to train on truth, and then they're going to allow us to extrapolate from a small number of truths to a much bigger number of truths. And that to me is where these models will become really powerful. But from where I sit in my seat at Bristol Myers Squibb, what I have to be careful of is over-investing, or there's also a risk of under-investing in any of these technologies. And so if you believe you're on the slope of enlightenment and you're sort of fairly far along, that's a great place to say we're going to increase our investment there. And we're thinking a lot about molecule invention as being part of that slope. If you say we're kind of really early, not quite in the trough of disillusionment, but you're not too far from it, and I think causal human biology is one of those areas, you have to be much more cautious about how you invest. If we look back in 3 or 4 or 5 years, we can look back on those statements and we can say, was I right to underinvest in one area or overinvest in another? And I'm sure many, many people are thinking about the same types of things.
Yeah, it's constant learning, isn't it? Well, I have to say, your blog has had a profound impact on our industry because you have an incredible skill at framing the problem and framing how we go about finding the solutions and stepping through these incredibly complex issues. So first, congrats on the blog. I think a lot of people all over the world read it. Let's pivot a little bit to another skill of yours, which is leading teams and how you think about leading teams, how you think about building talent, some of these aspects that really are very important in addition to all of what we've talked about because of the journey and the difficulty.
Yeah. Well, maybe I can start by just saying thank you to people that have been served as my mentors along the way. I sometimes go all the way back to high school. I had this great English teacher, Jack Labonte, who sort of taught me how to think and to write. And I don't thank him enough, but that was a really kind of important mentor for me. Fast forwarding to my PhD, I mentioned my PhD advisor, Hunt Willard, but it was this amazing environment. Aravinda Chakrabarty was there at the time, and it was this great environment at Case Western that I think was really impactful. And then David Altshuler at the Broad Institute was incredibly impactful. And then Rupert Vesey, who we know very well, is another individual who has really, I think, taught me a lot about industry, managing teams, how to think about teamwork. So I will describe some of my kind of lessons learned or some of the things that I apply, but I kind of want to start by saying I'm incredibly grateful to a number of mentors that I've had over the course of many years who have really kind of taught me many of the things that I now apply kind of in my job. So one, when I think about teams, you do need that North Star. So you need to have something that everyone is going to be able to rally around and say, that's where we want to go. And if you don't have that North Star, it's hard to basically build teams and to lead in a particular direction. So we always start with that North Star. So for us, making medicines, but not only making medicines, it's following these principles, quality, urgency, and efficiency, and being able to test the ideas really early in development. There are a few ideas that we've started to unpack there as well. So you got the North Star, and then, you have a group of people that kind of believe in that North Star. And, I'm fortunate where I am today at Bristol Myers Squibb. We have this amazing group of team members, not just my direct reports, but many of their direct reports who believe in the North Star and are working towards that goal. But then you need to make sure that people have this skill to lead a particular team, and then they've got sufficient clarity around roles and responsibilities that they know if they deliver on what they're asked to deliver, that they'll kind of be rewarded, but it'll also contribute to the bigger picture. And then for me, providing the sufficient direction, but also the sufficient independence to say, here are the objectives, here are some constraints, but really work really hard to deliver on those objectives. And if you do, not only will you win, but your peers will win and we'll all win as a team. And that feels like, we're all now making progress towards that North Star. So providing sufficient independence to work towards that, but also being comfortable with a certain amount of uncertainty, because you can go too far on defining constraints or too far on defining roles and responsibilities. And now you're beginning to take away creativity from folks as well. So you need a little bit of uncertainty, a little bit of ability to pursue things that fall outside of kind of normal boundaries or being opportunistic. But as long as people are trying to do the right thing towards that North Star, I think you can put together kind of teams that are incredibly impactful. And so given the magnitude of the problem, I talked about those 1,000 steps along the way, you need big teams in order to accomplish this. So it won't be, individuals with deep expertise in one and only one area that won't try to stitch it all together. So it's big picture, teams, roles and responsibility, some ability to be creative and flexible, and then just everyone saying, we're in this together. We're all going to try to do the right thing to advance our mission. And I think we've done a pretty good job of that here.
We've talked about how difficult it is to find the wins. That means by default, we're dealing with a lot of failure. You've talked about uncertainty. A lot of projects, unfortunately, at some point on the journey fall by the wayside. How do you think about failure and the resilience to get up and go again?
Yeah. So I think we define failure as success or not of a particular program. I think we should probably redefine failure.
Yeah.
And it's, did you make the right decision? Mm-hmm. And then the outcome is, so in some ways, independent. But if you do everything to make the right decision, then whatever the outcome, it's the outcome. Yeah. And so it could be a quote unquote success or quote unquote failure, but it's not the outcome that's important. It's the decision leading up to the outcome. So just quickly as a pivot to poker, you can make, bets, and you can make bets that are very much the right bet to make at the time, and you can still lose the hand. Mm-hmm. It doesn't mean that you failed because you lost the hand. It just means, sometimes things don't break in your direction. So I try to redefine it not as success or failure, because then I think it puts too much emphasis on the outcome, but rather probabilities. Did you make the right decision? And then being accepting of the decision that occurs. So rather than saying, did a drug succeed or fail? Did you make the right decision? And then if you make the right decision, if it didn't work, that's okay.
Yeah.
That's okay. So that's not a failure, even though somebody might look, an analyst might say, the drug failed. And that's a true statement. The drug did fail. But the decisions were all the right decisions leading up to that particular, and that's all we can do. Yeah. So I try to refocus people on not the outcome, but the decisions, and trying to think not deterministically, but probabilistically. And then sometimes I try to use the poker kind of analogy, and works for some and it doesn't work for others. But I think thinking probabilistically is really important.
Yeah, totally. I love the fact that you position success or failure as in the context of this decision science that I think we're tackling here. All of your prior comments about just how little we know. I think Roger Perlmutter was the one who famously said it's a bloody miracle that we have any drugs given this complexity. Yeah. And so it is all about learning, isn't it? It's about learning, dealing with the data you have, and making the right judgment call about whether to continue investing. Are we actually helping patients, or are we taking resources away from another project where we might be closer to helping a patient?
Yeah.
So, all right. So maybe we can change direction now to learn a little bit more about you.
Okay.
What inspires you beyond the work of helping patients and helping teams to progress this difficult journey of making drugs. What about you as a person? What inspires you? What do you enjoy doing? Things that people wouldn't necessarily know about you.
So within work or outside of work?
Both.
All right. Let's see. So within work, I think we've talked a lot about the topic, which is a big idea, defining the catalytic steps, working towards that, doing that with principles in mind, and with the right people and the right kind of culture and motivation. So that I love doing. And I think that's one of the things that I love about my job in industry is, I have the privilege of being able to do that. Outside of work, I've got an amazing wife who is incredibly supportive, and we talk about so many things together, both professionally and kind of personally, that we spend just a lot of time together. And it's not like we do one thing like, golf or tennis. She's way better at me than tennis. I'm not particularly good at golf, so that's not even a particularly a good example there. But we just do a lot of things together. I also have three daughters who are now young adults, and it's been wonderful to be part of their journey. Mm-hmm. And so my oldest daughter is doing research in a lab at Penn in cardiomyopathy research. And so I love just talking with her about some of the things that she's working on. My middle daughter is a junior at Tulane. She happens to be doing a semester away right now. And she's interested in science, but also the intersection of science and business. So I love talking with her about things that are of interest to her. And then my youngest daughter is a senior in high school, and she's thinking about where she wants to go next year. And And she's got all sorts of interesting ideas as well. So it's less about me and my interesting idea, but trying to connect with people and what they're interested in, and then finding ways in which we can kind of have that shared kind of experience together. So for each of my daughters, they're different people, and trying to understand what motivates and gets them excited. And I mentioned my wife as well. But I also have a bunch of great friends. We like to do things together, whether it's, golf or tennis or hikes or just kind of socializing, occasionally playing poker. My parents are amazing, and they're in their 80s. My dad just turned 90. And so we spend time as a family, and a lot of times it's just hanging out and talking. And then my sister and her family are also amazing as well. So I'm really, really fortunate to have interesting, thoughtful, warm, caring people around me, and I just like spending time with them. It's, I guess, not like I've solved these feats of music or hikes or athletic accomplishments, but it's just kind of like the fundamentals of people. I like that a lot.
Wonderful. I'm interested in the comment about your daughter's interest in science and business. This idea of convergence between disciplines fascinates me. My son actually is similar. He has a passion for science, but he's also super interested in business. Convergence, I think, is the disciplines, physics and biology coming together. Any commentary there on the future young generation and how they should be thinking? We talked about domain expertise very early on.
Yeah.
When you think about the next generation and the types of things that they should be embracing, at least being open-minded to.
I like the ability to go deep in a topic, but also the ability to go broad in a topic.
Yeah.
So there is depth and breadth to most of the problems that we try to solve. And so if you can't go deep, I think you're going to be not very successful. But I also think if you can go deep but not broad, I also think you're going to have limited professional success. It doesn't mean that you won't be successful. It's just, I think, more limited. So I do really like the ability to say you can go deep in a topic, but then you can do it across disciplines, and then you can figure out how to kind of stitch things together. So, sometimes I'm asked, what advice do I have for young folks? Well, the first thing I always say is beware of the personal narrative. Old people us, will give advice, and then you have to unpack what really happened, way back when, because they often are giving you advice through this retrospectoscope, which probably isn't quite as accurate as people think that it is today. So beware of the personal narrative. Pick something, anything that you're interested in, go deep. But then to your point, figure out how it all connects. Be able to go broad and stitch things together. Because I think there are people who can go incredibly deep but can't go broad. But if you can figure out how to stitch them together, I think that's where a lot of special magical things happen. And we talked about the 1,000 steps in drug discovery and the fact that it's a miracle that anything ever gets approved. There's a lot of breadth in that process. If you just want to do human genetics, you're not going to be successful in drug discovery and development. You got to go across multiple different areas. And fortunately, there are so many fascinating things about what we do: the regulatory science and formulation science and operational science and commercial science and how to actually position medicines. But you got to be prepared to go deep and broad. So stitching a lot of things together is really important. Kind of forgot the original question, so I probably didn't even answer your original question.
You did. You definitely
another LBL. I'm sorry.
No, not at all. So as we come towards the end here, we talked about your early career working with patients in RA, I believe. I think, and maybe you can tell us, that the life of a patient with those types of inflammatory diseases has changed pretty profoundly over the last 20 or 30 years. Maybe we can use that as a moment of inspiration for what the next 20 or 30 years will hold. Maybe just looking back, what kind of cures, treatments people have, and how that sets us up for the future.
So you may not know this. This is one of my favorite questions.
Okay.
So, and I not only is my favorite question, is that we increasingly have an answer to the question, which is totally exciting.
Indeed.
So I finished my MD, PhD in 2000, and I started my internal medicine residency and my rheumatology clinical training in about 2002. So the reason that moment in time is important is this is when all the anti-cytokine therapies, the anti-TNF therapies will really begin to make their way into clinical practice. And so my mentors in clinical medicine would say, the waiting rooms in rheumatology in the 70s and 80s and even 90s look very different than they do today in the 2000s. And there was a lot more disability pre-2000 than after 2000 because of these medicines. So suddenly the waiting rooms looked more like just a normal waiting room of patients going for primary care visits because of these medicines. So what the medicines did was they would control inflammation to prevent a lot of the disability in rheumatoid arthritis and lupus, et cetera. But what they didn't do is cure disease. So I would have conversations with patients in the early 2000s, and I would say, we understand what your disease is. We have a therapy to intervene and treat that particular disease. So your symptoms will get better, but there is no cure. So you're going to have to be on this medicine, which often is a powerful immunosuppressive medicine for the rest of your life. So it'd be this sense of relief when I would sit across from a patient to say, this is what I have, I've got a treatment. But there would be this sense of disappointment when they realize there is no cure and I'm going to have to be on these strong medicines for the rest of my life. Like, that's really hard for a person to accept.
Yeah.
So that's when I began to think there's got to be a way to have cures of these different autoimmune diseases. And we put together kind of this model, and Dennis Zoller well.
Yes.
Dennis was a real inspiration for me in terms of thinking about this concept of sequential immunotherapy, to say you can control inflammation, but can you also reset immune memory? So Dennis had these ideas many years ago of resetting immune memory. And you can kind of build on what we know from hematopoietic stem cell transplants and some other experiments of nature to say it's possible, but how do we do it pharmacologically in a way that can actually get to a large number of patients? So we started to think about different ways in which that could actually be done. Fast forward to where we are now, and it looks like a fairly simple approach, which we sort of underappreciated at the time, of CD19-based B-cell depletion can reset immune memory in patients with autoimmune diseases, lupus, potentially RA, myositis, scleroderma, maybe multiple sclerosis, or kind of a long list of them. And we're seeing evidence of that with this approach with kind of autologous CAR T. But it's a pretty cumbersome process that's not going to get to nearly as many patients that deserve to have this intervention. But at least it shows that it's possible.
Indeed.
So I believe we're now, if I go back 25 years, anti-cytokine therapy, it changed the waiting room of patients in rheumatology offices. Today, I think we're also at the very start of this amazing journey of being able to reset immune memory so that patients can come off of all medicines. So not only are they controlled in terms of their inflammation, they're not getting the joint destruction and other organ destruction from chronic immunosuppressive therapies, but they're now going to be able to go off all of medicines and lead a totally normal life. I mean, think about it as antibiotics for an infection. The infection clears and you no longer have to worry about it. Like, is that possible? And I think that it is.
It's very inspiring to watch the data coming out of these trials. I saw the Cabaletta data recently. I mean, incredible. These are cures, essentially, as you're pointing out. And so we have the opportunity, I think, to look forward over the next 20 or 30 years to when this becomes not just validated in a broader population, but the mechanisms required to get them to a broader population. That's, I think, some of what we're going to solve in the future.
Yeah. So it's totally exciting. It's rooted in, fundamental scientific discovery, thinking about patients, working towards that North Star of making medicines for patients. And I think, at least in the case of rheumatologic diseases, I think we're on the cusp of a second revolution. It's
been absolutely terrific to spend this time with you. I love your LBLs. I think we've now just...
Can you tell my kids that, please? That would be great.
Yes, and hopefully we'll get a lot of feedback from this incredible insight that you've provided about making medicines. It's been awesome. Thank you.
Well, Karen, so fun. Thank you. And it's been great to know you for so long and to have these conversations at dinner, over coffee, and now kind of recorded via podcast. So thank you.
Indeed. Yeah. See you soon.
All right.